Walk into almost any supermarket and place 6 vegetables beside one another. A bunch of curly kale, a round head of cabbage, a thick stalk of broccoli, a white cauliflower, a tower of Brussels sprouts, and a strange, swollen kohlrabi. They look as if they belong to completely different parts of the plant kingdom. One is a leaf, one resembles a miniature tree, one looks like a pale brain, one grows as dozens of tiny green balls along a tall stalk, and one appears to be a turnip that somehow forgot to grow underground. But botanically, they are all the same species, Brassica oleracea. Every one of them was created by human beings repeatedly choosing which part of a single wild plant they wanted to enlarge. Choose the leaves, generation after generation, and you move toward kale. Choose the tightly packed central bud, and you create cabbage. Choose the side buds, and you eventually get Brussels sprouts. Choose the stem, and the plant becomes kohlrabi. Prevent those flower structures from developing normally, and the result becomes cauliflower. This was not done in a laboratory. There were no microscopes, genetic tests, or written breeding plans. It was done slowly by farmers saving seeds from plants that looked slightly more useful than the others. And across centuries, those tiny choices pulled one wild plant apart and rebuilt it into some of the most recognizable vegetables on Earth. But the story begins before any of those vegetables existed, before the round cabbage head, before broccoli, before cauliflower, before anyone had ever heard the name Brussels. It begins on a cliff. Along parts of the European coastline, a rugged plant grew where many others struggled to survive. Its roots clung to rocky ground. Cold winds battered its leaves. Salt spray blew in from the sea. The soil was thin, alkaline, and exposed. The plant that survived there did not resemble the neat cabbages found in modern fields. Wild cabbage grew as a loose collection of thick, waxy leaves around a tough stem. It needed those heavy leaves because life on a coastal cliff was difficult. The leaves helped retain water. Their waxy surface reduced damage from wind and salt. And the plant's ability to tolerate lime-rich ground allowed it to occupy places where competing vegetation was limited. It was not beautiful, it was not especially tender, and it had absolutely no intention of becoming 6 different vegetables. Its only purpose was survival. Exactly where domesticated Brassica oleracea first emerged remains surprisingly difficult to prove. For generations, botanists assumed the wild cabbages found along the coasts of Britain and Western Europe were the direct ancestors of the crops. Modern genetic research complicated that picture. Some supposedly wild populations may actually be descendants of cultivated cabbages that escaped gardens and returned to the coast. Other evidence points towards Mediterranean relatives and suggests that domestication may have followed more than one lineage. Even after centuries of study, researchers are still untangling where the wild plant ended and the cultivated plant began. But the larger story is clear. Somewhere around the Mediterranean world, people began gathering and cultivating leafy cabbages thousands of years ago. And once farmers saved seeds, natural survival was no longer the only force shaping the plant. Human preference entered the story. The earliest growers were probably not trying to invent something revolutionary. They were looking for dinner. A bunch of slightly larger leaves gave more food. A plant with less bitterness was easier to eat. A plant that survived winter was worth planting again. A plant that produced tender shoots was more useful than one with tough, fibrous growth. So farmers saved the seeds of those plants. The following season, they repeated the process. Then their children repeated it, and their children after them. Nobody knew what a gene was. Nobody understood mutation, inheritance, or chromosomes. But the farmers were changing inherited characteristics each time they decided which plant would reproduce. Nature created variation. Human beings decided which variations would continue. That process is called artificial selection. It uses the same raw material as evolution, but instead of the environment alone determining success, human beings choose the winners. And cabbage would become one of the most spectacular demonstrations of that power. By the time of ancient Greece and Rome, cultivated forms of cabbage were already garden vegetables, but they were generally leafier and looser than the tightly packed supermarket cabbages we recognize today. Greek and Roman writers described different forms of cabbage, and Romans ate leafy brassicas both cooked and raw. They could preserve vegetables in brine or vinegar, allowing them to last beyond the harvest season. To Roman society, cabbage was not merely food. It developed a reputation as medicine. The Roman statesman Cato the Elder distrusted many foreign medical practices. He preferred simple remedies connected to traditional Roman life, and few things impressed him more than cabbage. Cato credited it with an almost absurd range of healing powers. Roman and Greek traditions recommended cabbage preparations for digestive trouble, wounds, headaches, and other conditions. Some people even believed eating cabbage could reduce drunkenness, perhaps because the plant was imagined as the natural enemy of the grapevine. These claims were not based on modern clinical science, but they reveal how deeply the plant had entered ancient life. Cabbage was cheap enough for ordinary meals, respected enough to appear in agricultural writing, and powerful enough in the Roman imagination to function as a near-universal remedy. Yet the cabbage eaten in ancient Rome was not the end of the plant's transformation. It was the beginning. Farmers across different regions began emphasizing different parts of the same basic plant. And this is where the cabbage family becomes genuinely strange. Imagine the wild cabbage as a set of possibilities. It has leaves, it has a central growing point, it has side buds, it has a stem, and eventually it has flowers. A breeder could favor any one of those structures, and depending on that choice, the descendants would move in completely different directions. The oldest looking path was the leaf. Farmers who repeatedly saved seeds from plants with the largest, most tender, or most curled leaves developed forms resembling kale and collard greens. Kale does not need to produce a compact head. Its value lies in the leaves themselves. The plant remains open, allowing growers to remove leaves gradually while it continues producing more. This makes kale look closer to its wild relative than many of its unusual cousins. But it is not untouched nature. Its size, texture, color, cold tolerance, and leaf shape are all products of selection. Humans had taken the wild plant's ordinary leaves and exaggerated them. Then another group of farmers made a different choice. Instead of selecting plants whose leaves spread outward, they favored plants whose inner leaves crowded around the central growing point. Each generation, they saved seeds from plants with tighter centers. The leaves folded inward, the gaps became smaller, and over time, the loose coastal plant transformed into a dense ball of overlapping leaves. The cabbage head was born. What we call a cabbage is essentially an enormous terminal bud. It is the plant's growing point trapped inside layer after layer of leaves selected to wrap around one another. Cut the head open, and the entire history of that selection becomes visible. The outer leaves are broad and protective. The inner leaves become increasingly pale, compressed, and tender because sunlight struggles to reach them. Humans had turned the center of the plant into a living storage chamber. This dense structure changed what cabbage could do for people. A leafy plant is useful during the growing season. A compact cabbage is useful after it. Its tight structure helped protect the inner leaves and made headed cabbages easier to harvest, transport, and store. In cool conditions, some varieties could remain usable long after more delicate vegetables disappeared. That made cabbage especially valuable in Northern Europe, where winter reduced the availability of fresh produce. For poor households, it offered something reliable. It could be boiled into soup, cooked with grain, added to stews, combined with meat when meat was available, or eaten with little more than salt and fat when it was not. Cabbage did not become a staple because it was luxurious. It became a staple because it refused to be useless. It tolerated cold. It produced a large amount of edible material. It could fill a pot, and almost every part could be consumed. By the Middle Ages, cabbage and other brassicas were deeply established in European agriculture. They appeared in household gardens, monastery plots, and the fields surrounding villages. It was not the food of a single class or region. Its many forms moved through Europe because they could be adapted to different climates, soils, and culinary traditions. But headed cabbage was only one possible answer to the question, what part of this plant can humans enlarge? In southern Europe, especially around Italy and the Mediterranean, growers increasingly focused on flowering structures. Normally, a cabbage family plant eventually sends up stems carrying many small yellow flowers. But farmers selected plants with thicker, more abundant shoots and clusters of unopened flower buds. Over many generations, those shoots became broccoli. The word itself comes through Italian and refers to the flowering crest or sprout of cabbage. And that description is exactly what broccoli is. When you eat broccoli, you are eating a thickened flowering stem topped with hundreds of buds that have not yet opened. Leave broccoli in the field too long, and the tight green head begins loosening. The buds separate. Yellow flowers appear. The vegetable reveals what it had been planning to become all along. Broccoli's development is closely associated with Italian cultivation. Forms of flowering Brassica were known in the ancient Mediterranean world, and Italian farmers refined them over centuries. Different regions produced different sprouting types. Some formed one dominant head, others produced many smaller shoots. What modern shoppers call broccoli is not the inevitable form of the plant. It is a frozen moment in its reproductive development. Farmers decided that the plant was most valuable immediately before flowering, and then bred it to remain in that edible stage longer and produce more of it. Cauliflower pushed this same idea even further. Broccoli still looks like a collection of recognizable flower buds. Cauliflower does not. Its pale head, known as the curd, consists of a densely compressed mass of undeveloped flowering tissue. The developmental process has been interrupted and exaggerated until the structure folds into itself. Instead of extending into normal flowering branches, the plant creates a compact surface of repeated, tightly packed forms. This is why cauliflower can resemble a brain, a cloud, or a coral reef. It is a flower system that has been prevented from behaving like ordinary flowers. Early cauliflower history is difficult to separate completely from broccoli and other flowering cabbages. Medieval sources associated important forms with Cyprus and the Eastern Mediterranean. Seeds later moved into Italy and across Western Europe. By the early modern period, cauliflower was becoming a prized garden vegetable, though it remained difficult to grow.
The curd could be damaged by heat, sunlight, poor soil, or bad timing. Growers often protected the developing head by folding leaves over it, preventing sunlight from discoloring the pale surface. Unlike the humble field cabbage, successful cauliflower announced skill. It demonstrated that a gardener had controlled soil, temperature, moisture, and timing well enough to produce something unusually delicate. One species had now become both the food of peasants and the achievement of expert gardeners. Then there was the stem. Most people looking at kohlrabi assume it is a root vegetable. It resembles a turnip sitting partly above the ground. But the swollen sphere is not a root. It is the stem. Farmers selected cabbage relatives with increasingly thick stems until the section above the soil expanded into a crisp, edible ball. The leaves still emerge from its sides, making the plant look like a vegetable from another planet. Its name reflects the confusion. Kohl refers to cabbage, while rabi relates to turnip. In other words, it is the cabbage that looks like a turnip. Again, no new species had been discovered. Humans had simply taken a supporting structure and made it the main event. The most visually bizarre transformation may have occurred in the Low Countries. Look closely at an ordinary plant and you will find small buds where leaves join the main stem. These lateral buds can potentially develop into branches. Growers near what is now Belgium selected plants with unusually enlarged buds running along the stalk. Generation after generation, those buds became tighter and rounder. Eventually, one plant could carry dozens of miniature cabbage heads. These became Brussels sprouts. They were cultivated in the region around Brussels and became especially associated with the area. The name stayed attached to the place that helped make them famous. A Brussels sprout is therefore not a baby cabbage harvested early. It is an enlarged side bud. A full plant can rise from the ground like a strange green tower with sprouts arranged along its stem. The resemblance to cabbage is real because the same basic selection has occurred at a different location. Cabbage enlarged the terminal bud at the top. Brussels sprouts enlarged the lateral buds along the sides. It is the same biological machinery redirected by human choice. By this point, the transformation was extraordinary. From one species, farmers had created vegetables centered on nearly every major above-ground structure. Leaves became kale, the central bud became cabbage, side buds became Brussels sprouts, the stem became kohlrabi, flowering shoots became broccoli, compressed flowering tissue became cauliflower. And those are only 6 of the most famous forms. Collard greens, Savoy cabbage, sprouting broccoli, and many regional varieties reveal that the species contains far more variation than a supermarket display suggests. The plant is not really a collection of 6 vegetables. It is a biological construction kit that farmers have been rearranging for centuries. Charles Darwin found domesticated variation deeply important. When explaining how selection could reshape living organisms, he studied the dramatic differences created under human control. Domestic crops and animals demonstrated that small inherited changes accumulated over generations could produce forms that appeared astonishingly different from their ancestors. The cabbage family offered visible evidence that one species could contain enormous potential. Humans did not create variation from nothing, but by repeatedly preserving certain traits, they could push that variation in directions nature alone might never have favored. And farmers were not selecting only for shape, they also changed taste. Wild brassicas defend themselves with chemical compounds called glucosinolates. When plant tissue is damaged, these compounds contribute to the sharp, bitter, mustard-like flavors associated with cabbage, kale, broccoli, and Brussels sprouts. The chemicals help discourage insects and other animals from eating the plant. But humans have a complicated relationship with bitterness. A small amount can taste interesting, too much can make a vegetable nearly unbearable. So breeders balanced 2 competing demands. The plants needed chemical defenses, but people wanted them to taste better. Across generations, farmers altered bitterness, tenderness, sweetness, color, and smell, along with physical shape. This process continues today. The Brussels sprouts many people remember from childhood were often strongly bitter, especially when boiled until soft. In the late 20th century, researchers identified compounds associated with undesirable bitterness. Breeders then used older, milder varieties to help develop modern plants that combined improved flavor with good yield and disease resistance. The result was a new generation of Brussels sprouts that many consumers find sweeter and less harsh. Cooking methods changed as well. Roasting encouraged browning and crisp edges instead of the sulfurous smell and soft texture produced by prolonged boiling. The vegetable did not suddenly become fashionable by accident. Human beings had modified its flavor again, just as earlier growers had modified its size of buds. Cabbage itself was also transformed by something farmers could not see: microbes. Cut a cabbage, add salt, press it beneath its own liquid, and naturally occurring lactic acid bacteria begin changing the leaves. The microbes consume sugars and produce acids. The environment becomes hostile to many organisms that otherwise would cause spoilage. The cabbage softens, its flavor becomes sour, and the food can survive far longer than fresh leaves. In Europe, fermented cabbage became sauerkraut. In other places, different cabbage relatives and seasonings created other fermentation traditions. The methods were not identical, and Chinese cabbage, used for many forms of kimchi, belongs to a related species rather than Brassica oleracea.
But the principle was similar. Humans recruited microbes to preserve a fragile harvest. Fermentation made cabbage especially valuable during winters and long journeys. Fresh vegetables disappeared quickly. Fermented cabbage could remain available when ships, armies, and isolated communities needed plant food most. During the Age of Sail, scurvy devastated crews deprived of fresh sources of vitamin C. Sailors did not yet understand vitamins, and proposed remedies were inconsistent. But fermented plant foods, including sauerkraut, formed part of attempts to protect crews on long voyages. The history is more complicated than the idea that one captain or one food solved scurvy overnight. Still, cabbage became part of humanity's gradual realization that disease could result not only from poison or infection, but from something missing in the diet. Cabbage crossed oceans with migration, colonization, trade, and war. European settlers carried seeds to new climates. Immigrant communities carried recipes. A vegetable associated with necessity in one country could become cultural identity in another. It appeared in stuffed leaves, soups, pickles, dumplings, stews, salads, and fermented dishes. It absorbed the flavors of whatever surrounded it: vinegar, pork, grain, chilies, garlic, cream, spices. Cabbage itself was rarely the most expensive ingredient. That was precisely its power. It could stretch richer foods across more people. It could turn a small amount of meat into a full pot. It could provide bulk when grain was scarce. It could preserve the memory of home after families moved thousands of miles away. In modern agriculture, the family continues to change. Plant breeders search for resistance to pests, heat, drought, and disease. They select crops that mature uniformly for mechanical harvesting. They develop broccoli with longer shelf life, cauliflower with new colors, and cabbages suited to different seasons and processing methods. Seed banks preserve old varieties and wild relatives because the traits rejected by earlier farmers may become essential in the future. A tough, bitter plant may resist an emerging disease. A small coastal population may tolerate salt. A traditional variety with poor commercial appearance may survive heat that destroys modern fields. Genetic diversity is the reserve from which the next vegetable may be built, and modern genomics has revealed just how complicated that reserve is. The transformation from wild cabbage to kale, cabbage, broccoli, cauliflower, kohlrabi, and Brussels sprouts did not involve one simple switch. Researchers find changes involving flowering, leaf growth, stem development, hormones, and many interacting regions of the genome. Different growers sometimes selected similar traits through different genetic routes. Crossing between cultivated plants, wild relatives, and escaped populations further blurred the family tree. The vegetable aisle may look neatly organized. Its genetic history is not. It is a web of migrations, accidents, mutations, crossings, and human preferences accumulated over centuries. That is what makes the cabbage story more important than it first appears. We often imagine domestication as a single event. A wild plant is discovered, people begin farming it, and the modern crop appears. But cabbage shows that domestication does not have a clear ending. Humans continue reshaping plants every time they save one seed instead of another. Every time they choose sweetness over bitterness, a larger leaf over a smaller one, a delayed flower over an early flower, a compact head over an open plant. The vegetable keeps changing because human needs keep changing. The first coastal cabbages needed to survive wind, salt, and rock. Ancient farmers wanted edible leaves. Northern households wanted food that survived winter. Italian growers favored flowering shoots. Belgian farmers enlarged side buds. Other Europeans thickened stems and compressed heads. Modern breeders want disease resistance, uniform harvests, longer storage, and improved taste. The plant has never stopped answering human demands. And perhaps that is the strangest part. Kale, cabbage, broccoli, cauliflower, Brussels sprouts, and kohlrabi can still produce similar yellow flowers. Strip away the shapes humans exaggerated, allow them to flower, and their family resemblance becomes much clearer. The yellow blossoms reveal what the dinner plate hides. These vegetables were never truly separated by nature. We separated them with preference. A wild plant growing above the sea offered humans a collection of ordinary structures: leaves, buds, stems, and flowers. We took each one and pushed it toward an extreme. We made the leaves larger, the center tighter, the side buds rounder, the stem thicker, the flowering shoots heavier, and the undeveloped flowers denser. No single farmer planned the final result. No ancient civilization drew a picture of all 6 vegetables and decided to create them. Each generation made small choices for immediate reasons: more food, better storage, softer texture, improved flavor, a stranger appearance, a higher market price. But those small decisions accumulated until one species looked like an entire produce section. The history of cabbage, therefore, is not only the history of a vegetable. It is the history of human selection. It shows that agriculture did more than help people grow food. Agriculture gave human beings the power to redirect evolution. The wild plant survived by adapting itself to the cliffs. The cultivated plant survived by adapting itself to us. And today, millions of people eat its leaves, stems, buds, and flowers without realizing they are all eating different versions of the same ancient survivor. Not bad for a weed that once clung to the edge of the sea. If this story changed the way you look at the vegetable aisle, subscribe to Food Through Time for more hidden histories behind the foods that shaped civilization. And tell us in the comments which member of the cabbage family surprised you the most.